Change the title because Joe Jankowski preceded me. This is my obligatory disclosure information, which is that I was a founder and officer of Osiris Therapeutics, and you'll find out why that's important as we go on with this lecture. I'm currently divorced from Osiris. That means lawyers and litigation and bad feelings, so we can discuss that in a theoretical way also. I do receive, I'm not a consultant for Osiris, but I steal their public data to support some of my fancy ideas. I do receive royalties through Case Western Reserve University, who receives money from Osiris. I have other consulting activities that's unrelated to this lecture. So this is who I am. I'm born in Chicago, had my education at an engineering school, got my PhD at Hopkins, and in 1969 I came here, and I've been here ever since. So you can make two conclusions from that. One is I'm really not movable, or actually I like it here. And so it turns out both are true since my grown children have moved back to Cleveland. I'm no longer mobile, that's clear. So, and after spending over 40 years here and being hugely entrepreneurial, there's a lot of lessons that I've learned, and one of the lessons is how to give a really good lecture. To give a really good lecture, you have to have a good beginning and a really good ending, and you have to keep the two as close together as possible. So I stole that from George Burns. But I have a long story to tell you, and because of the audience is predominantly young people, I have some missionary work to do to try to tell you what you're going to see as IP lawyers or litigators who in the biological space. And so I'm going to talk about the area that I'm doing research in, and there's some very important lessons. And so the real title of this talk is the science and clinical use and patents, how they arose, what they mean, and what their context is, and about adult mesenchymal stem cells, and I'll tell you what those are in a few minutes. For a scientist, for us to take something from the bench to the bedside is awesome. I mean, we really feel fulfilled in a societal and personal sense. It turns out that I'm going to tell you that this technology that we started a long time ago, we took it from the bench to the bedside. And now actually we have to take it back to the bench because what we learn from human clinical data is completely unpredicted in the early parts of this technology, and therefore the whole patent structure needs to be rethought about and is actually going to cause the current patent holder some severe problems. So, and I'll try to point those out. So the new use of the same old thing makes the whole legal issue complicated. So this is probably the most important slide you're going to see, so you need to memorize this, which is if I discover something in my lab this afternoon, it takes 20, and I write a paper, it takes 20 years minimum to get that into the marketplace. So if you have some bright idea now, and you have to understand 20 years. This is for light bulbs, TV sets, and all of these biological materials that I have listed here, recombinant proteins, monoclonal antibodies, antisense, stem cells. And so in 1990, the 4 basic patents for mesenchymal stem cells were written and were filed. And the first product was approved. It turns out it's in Australia, it's not in the States. The first product based on this technology, not on those patents, based on this technology, was in Australia in a company called Mesoblast in 2010, 20 years. So it turns out 20 years is the minimum. So you have to understand that patenting, licensing, clinical trials, all of these are huge, timely, time-dependent processes, and you have to be sensitive to that in all of these considerations. So I'm going to talk about this completely new area, which is called regenerative medicine. Regenerative medicine is the concept is to replace, repair, maintain, add value to organ function that's lost or compromised through injury or disease. And there's two kinds of regenerative medicine, and they're very distinct and they're very different. One is pictured on this slide, which is called tissue engineering. You take cells that make tissues and you put them in a scaffold in culture, in vitro, and then you surgically implant them in the body. So that's tissue engineering, and I'll show you some examples of that. The other kind of regenerative medicine is actually a very old idea, dates back to the Greek god Prometheus, where he had some organs that were the other gods didn't like him so much, so they sent this eagle, this hawk, to eat away at his liver, and his liver regenerated. So it's actually this concept that you can regenerate yourself. And that, it turns out to be a central biological fact. You are regenerating yourself as you sit here and try to decide whether you're going to be interested in what I'm saying. And so I'm going to show you a video of a newt that's going to regenerate its limb. So we cut off the limb, and this is an actual time-lapse picture. And what you see is this limb is totally being regenerated. And this is an animation of this process so that I can break down the details for you. So there's the limb as you start, quite flexible, and a scissors is going to come in and snip away the arm, but it could do it at any place. You see a bleed, you see a scab form, and the ectoderm, the outer layer, covers it. And now we're going to go into this limb and look at the action. So here you have the cut surface, and these cells are the stem cells that are aggregating here to form what's called a repair blastema. And from this wad of cells, you're going to see the formation first of a very small limb, and then that limb is going to grow and regenerate perfectly. So if you cut the limb off here or here or here, the information involved in what's left sends a message so that it's a perfect regeneration. And so I would contend that you could do that if you were 1 or 2 years old. And as a matter of fact, there's a literature on finger guillotining of young people. The data comes from England, and there's a key piece to their regeneration, including the first 2 joints. So you would have said, because you learned in school you can't regenerate anything, right? You would say you can't do that, but these kids did it, and it's documented. There's several important rules. What you saw on the newt was that there wasn't an emergency room. There was a bleed, there was a scab that formed, and there were no stitches through that wound. You throw the animal back in the pond, dirty, horrible pond, as a matter of fact. And so that's the first rule of digit regeneration. These kids were all in rural England, and there was no emergency room or these shopping mall health centers to throw stitches through it. You put a stitch through that wound, that's it. It ain't going to regenerate, because that outer covering that I showed you is the key to the successful outgrowth. So what the parents did is put a clean cloth on it, got a nice scab to form, and then the kids, including the nail bed, regenerated these fingers. If you did that with a 5-year-old, it doesn't work. And it's the stem cell titer decreases as we get older. The old farts like me we're lucky we're breathing. And I'll show you data to support that. So I want to talk about a special kind of a stem cell called a mesenchymal stem cell that I named several years ago. And I want to, and again, I'm a missionary, so I'm going to do science on you. You're going to have to learn a little science. But I'm going to tell you how this relates to patents, because I think it's the key, the information is the key to the process. So you're alive because every second, 15 million blood cells drop dead and are replaced in your body. So while you're contemplating that, a couple of 100 million cells have dropped dead, and you have automatically replaced them. And the basis for that is in your bone marrow, there's a cell, a rare cell called a hematopoietic stem cell that gives rise to all of those cells that drop dead. So for example, the recombinant protein EPO generates part of that pathway and accentuates red blood cell formation. So if my name is Armstrong and I want to do a hill climb, I EPO up so that I have lots of red blood cells, more than normal, and I test it the night before by taking a blood sample and finding out that the cell count in my blood is gigantic, so I can go do the hill. That's called doping. So whether Lance wants to accept that or not, that was legal when he did it, I have to say. It wasn't forbidden at that point, but it was legally doping by manipulating this normal regenerative pathway to accentuate red blood cell formation. So it turns out in bone marrow there's another stem cell. That stem cell I have named mesenchymal stem cell. And the basis for calling it a mesenchymal stem cell is that it makes bone or cartilage or muscle or marrow stroma. Marrow stroma is this scaffolding in which all of hemopoiesis takes place in your marrow. So it's a cell which eventually makes the fabric to support all of this hemopoiesis, a tendon, ligament, fat, dermis, etc. So those are cells from the middle layer of an embryo. The middle layer of the embryo is called a mesodermal layer. So the fancy English term is mesenchyme. So I call these cells mesenchymal stem cells. And in the mid-'80s, the dogma of the day was that you had one stem cell, your hematopoietic stem cell, and that all of the other organs of your body, there were no stem cells possible. And the logic for that is you couldn't regenerate all these organs. And there was some data to support that. All that data turns out to be, the data is correct, the interpretation was not so correct. So this is a hypothesis slide for which there's data which I won't go through. And what this said is that in bone marrow, the mesenchymal stem cell, the MSC, if you learned how to talk to it properly, it could go down a pathway and make bone or cartilage or muscle, etc. Okay? So the basis, that's the basis for starting a company, which I called Osiris Therapeutics. And Case Western Reserve, in its infinite wisdom, transferred that technology to Osiris Therapeutics, and that's the basis for, since I'm just an employee of the university, I own nothing. So all of my intellectual property is owned by my employer. My employer is enormously generous, which is it splits 50-50 with the inventors, which is quite unusual in academia. It's a very high split, I have to say. So the university transfers the technology, which is embodied in 4 patents. And 1977, 78 was my divorce proceedings. We won't go into that. Osiris Therapeutics went public in 2006. Actually, this is the old logo of Osiris, and the CEO invented this logo, and he sees this as a cartilage defect in your knee that regenerates. Okay, he invented this on his yacht on a very wavy day, and I think he had a little too much to drink, but that's another issue. This is the new logo for Osiris, which you'll see later on. So the original MSC patents, which I'll show you in detail in a few minutes, focus on this multi-potency, the ability to go into these bone, cartilage, muscle avenues, and their use in tissue engineering to reform orthopedic tissues, for which there's some need, because the skiers among you know that you got a divot in your cartilage in your knee and you're in trouble because you cannot regenerate that. You break your bone, that heals, doesn't exactly regenerate, it heals. That's a different process. So The problem is all of those patents are based on this tissue engineering logic. Take the cell, expand it in culture, get enough of the cells, put them in a fabric, get them to be cartilage, put the wad of cartilage back on, and make sure it integrates with the host tissue. So I'm going to tell you the news story. So I'm telling you the end of this lecture first so that you can see what the problems are going to be. So it turns out that what we screwed up on in our early studies is we never asked, where are the MSCs? What are they doing naturally? We got them out, we put them on a petri dish, we thought that was pretty good, and we went on from there to study them and figure out how to control them. But what are they doing naturally? It turns out that all, this is dogmatic, so there's going to be some exceptions, but all MSCs are what are called pericytes. They sit on every blood vessel in your body. And they have a very precise function in these locations. This is actually a small capillary in heart tissue, and my friend Bruno Piot colored these cells red. They're not usually colored, okay? So, and they sit in tight association with those blood vessels and function in collaboration with the blood vessel. And when you get an injury, that pericyte comes off the blood vessel and it becomes an MSC. That MSC becomes activated by the injury environment that it's situated, and that MSC starts making molecules that it puts out into the local environment. Those molecules do two things. One is they put a curtain in front of the injured tissue and don't allow your overaggressive immune system to see the injury. This stops your immune system from setting up autoimmune reactions to small local injuries. The second thing it does, from its back, it puts out molecules which, because you have an injured or broken blood vessel, you don't have oxygen transport, nutrient transport, so the cells become deprived. Those cells normally see a deprived state and drop dead. These molecules produced by mesenchymal stem cells stop the cell from dying, from apoptosis, for the experts in the audience, and also stop scarring. Scarring is when a bunch of cells drop dead, your body has developed a way of filling the void with scar tissue. It's a quick heal way of keeping you out of danger. It doesn't bring back full function, but allows you to limp into the forest and not get eaten by the dragons. So it also brings in new blood vessels very rapidly. That's called angiogenesis. And actually, in all of your organs, you have in tissue intrinsic stem cells. So in your heart, for example, we were taught, when I was in medical school, we were taught you're born with a fixed number of cells in your heart, and they just get bigger and when you grow, but the number of cells is fixed, that there are no more cells. Well, that turns out not to be true. It turns out that when you take a female heart and transplant it into a male, you can tell donor from host. And when that donor dies, the hearts actually are sent back to a central processing facility, and we now know that new heart cells come from the host, not the donor. Host. And the data for that's very firm. So you have a chance, you nick your heart, you got a chance of filling the void with your own cells. Okay, so, and you're regenerating your heart, your liver, your muscle slowly, but you're regenerating them. And this is what this cell organizes. So that Prometheus slide is the way these cells can be used. And I would now, I want to change, so I've written an editorial in the Journal of Tissue Engineering to suggest that we keep MSCs because I'm known for MSCs. I don't want to lose my fame and fortune. So I've changed the name to medicinal signaling cell because it has nothing whatsoever to do with their stem cell property. Nothing. Zero. They're drug factories for sites of injury. Now you see the patenting problem. Same cell, completely different logic. They can be used both ways. The one that's coming to the clinic, I'll show you, is the medicinal signaling cell. So for immunoregulation and for these trophic activities, here's a partial list of all of the published and or clinical trials that are currently going on using MSCs. So for bone marrow transplantation, you can add MSCs. We thought they were making new scaffolding and helping the hematopoietic cells. Wrong. They're actually calming down the whole immunorejection of that system and adding drugs to the bone marrow transplantation. I'll show you data for that. Heart attack, infarct. The area that's infarcted doesn't get enough oxygen, becomes ischemic. The mesenchymal stem cells, if you get them there, will protect the heart from the degenerative aspects of a heart attack. So you can have a heart attack. Within 48 hours, you get mesenchymal stem cells infused. They can be from some other donor because they set up this blanket of molecules that don't allow your immune systems to see them. So Gabrielle's mesenchymal stem cells, of course I would prefer hers because she's significantly younger than I am, and her stem cells are a little more vital. If they're infused into my arm, not only would I not reject them, but they will go to the site of injury and do their magic, their drugstore magic. So stroke, same problem. You have this blockage of an artery or a vein, and you get these cells that don't get enough oxygen or nutrients. They drop dead. The mesenchymal stem cells, if you deliver them to that site, protect it. Spinal cord cuts. If you cut the spinal cord and you squirt in some mesenchymal stem cells, no scarring. The scarring is what inhibits the axons from regenerating across the scar. Lung, asthma. I'll show you asthma pictures. It's an inflammation. These cells calm down those kinds of inflammations. GI tract, Crohn's disease or inflammatory bowel disease, that's an inflammatory malady. And so you get huge steroid doses to try to calm down your immune system. The mesenchymal stem cells do the same. Acute kidney failure, regeneration of meniscus, tendon inflammation is a great story. You actually go to the website of a company called VetStem. They get from where the tail of a horse inserts, right in front is a big wad of fat. The veterinarian cuts the hide, takes the wad of fat, puts it in a FedEx package, sends it to San Diego 24 hours later from the fat, because the MSCs are sitting on every blood vessel in your body. Fat's a source of MSCs. So they send back a syringe with these MSCs. You squirt it into the inflamed tendon of the horse. 3 weeks later, it's back to normal. 5,000 horses have been treated, 79% perfect recovery rates. It's from the drugs. It's not making new tendon. It's not the tissue engineering piece. Diabetes, the cells that make insulin are attacked by your body. The MSCs stop that attack. Actually, and there's a grant to Osiris from the Juvenile Diabetes Association. They're running a clinical trial to prove that that's efficacious. Here, I sent that list to Osiris. I have nice relationships with them now, new management. And I said, okay, you have 40, 50 patents. Which of your patents are going to apply for these maladies? So, of course, they have an opinion. They have an opinion. They're not necessarily a court-proven right, but they have an opinion. So they put their patent numbers, which they allow me to show publicly because they're happy to show they have huge protection. It affects the economics of that. So for all of these particular issues, Osiris would say, we have patent coverage. So that's an issue or not. Here's a list: rheumatoid arthritis, lupus, MS, etc. And this is the only one they put patent coverage on. So let me say that, for example, I put here antibacterial. It turns out that these mesenchymal stem cells make a protein which is antibiotic. It kills bacteria. Here's the paper this is a fabulous basic research paper that just knocked my socks off, that these cells could make an antibiotic that could kill bacteria. And it explains a huge amount of actually our physiology, where we're getting lung infections all the time, and we thought, well, the immune system takes care of that. It just attacks, our immune system attacks the, but no. The damage that they cause, actually the MSCs in the neighborhood actually make a local antibiotic. If you get a huge dose of bacteria and they take over your lung, you have to supplement that with oral antibiotic. But your own body, your own cell is making this molecule which kills a number of bacteria, particularly those that are found in the lung, and we have some experiments that I won't discuss to use it. So is there a patent on the antibiotic properties of MSCs? For sure not. For sure not. So this whole patent portfolio that Osiris is sitting on, they were very clever not to put anything for antibacterial because they don't have squat diddly. So, and that, you could imagine this could be a big seller. So there is a new day. For those of you who go to work in IP firms, there's hope you'll have some jobs and some interesting patents to write based on this technology. And here's the complexity of all of that, which is the MSCs are not making one molecule to do these jobs. So I've just, you don't have to know the names or what these molecules are, but for the immunotherapy aspects, for the apoptosis, for angiogenesis, for all of these, there's molecules that have now been described and are in the literature that account for parts of those activities. But it's a multi-drug delivery vehicle. If you try to get a single drug through the FDA, it's a nightmare. If you do a combination drug, almost impossible. That's 2 drugs. Combination drug is 2 drugs. 3 drugs, forget it. So this is a multi-drug delivery vehicle. So you can imagine in the legal and pharma industry, this is going to be a walking nightmare for these people. And there will be no magic drug to take the place of the MSC for lots of these things. So this is the end of the story. So and I'll come back to the beginning so that you can see where all of this came from. But I've told you there's this pericyte that sits on your blood vessel. The pericyte comes off, is an MSC. That MSC can eventually actually make bone. It can differentiate in your body and make bone, and there's a pathway for doing that. Likewise, there's a pathway for this drug delivery aspect of it. Okay, and that's the new science for which there's only actually a very restricted patent portfolio. And I'll go through that. So now I want to take you all the way back, because the discovery process and then the subsequent patenting process is the story I want to tell. So now I've done my missionary work. If you memorized everything I said, you'll be at least 10 years ahead of the pack, because it's going to take 20 for some of this stuff to get out, so you're in good shape. But I want to go back and tell you about my history and how I got into this, because itself is an interesting story. So as a postdoc, I went to a lab of an eminent embryologist, and he was working with embryonic chick limb tissue. And what happens is that this is the leg of a 4.5-day-old chick embryo. And what we did was we cut that leg off, and we took the mesodermal cells, the mesenchymal cells, out of that leg and put them in culture and studied their differentiation into cartilage, bone, and muscle. And that's how I got into this whole business, working with these embryonic tissues. And so in the chick limb bud cultures, what we found was that there were these stem cells from embryos that made cartilage, bone, and muscle. Okay? So we actually, it's a long story, but we use these to get an assay of putting these cells in culture and taking an extract from bone and putting them on the culture and showing that there were some molecules in the extract which caused the cells to differentiate into cartilage. So we called that chondrogenic stimulating, chondrogenic is cartilage, stimulating activity. And we biochemists started to purify those molecules from bone to do that. Now, this is predicated on the work of Marshal Yours. Marshal Yours was an orthopedic surgeon. He took bone and he demineralized it. He took the calcium phosphate, for which most of you take calcium phosphate pills to try to put that mineral back in your bone. He took it out of the bone and he took that demineralized hunk of bone and shoved it into an adult mouse muscle pouch, and came back 6 to 8 weeks later and found that that sponge-like material had caused the formation in the muscle, in the most abnormal site in the muscle, of a huge hunk of bone. So he said there's some molecule in bone which causes the differentiation of cells into forming bone, and so he called the molecule bone morphogenetic, to make a morphology, morphogenetic protein, BMP. Okay? And for 20 years, he published over 300 papers on this technology. And if you have a bone break and it doesn't heal naturally, what they do now is they take out the tissue, the scar tissue that forms in the break, and they put demineralized bone chips there, because they have all the goodies in them to help stimulate the formation of bone and bridge that gap that you, for a variety of reasons, weren't able to do yourself. So that's a hugely interesting aspect. And so Yours did this experiment, demineralized bone into muscle, okay, and in this, and that's what caused us to try to isolate that magic molecule. We called it CSA, and my first 2 patents are on CSA, and the purification process, and then how to adapt that purified material to put it into back in to stimulate bone formation. So it turns out, I'm going to tell you that BMP has been cloned and is a recombinant protein, is now sold on the market. I'll discuss that. But I got a call from the company that did this and said, "Is your patent still active?" And I said, "You know, it's not my patent, it's Case Western Reserve's." It turns out Case Western Reserve failed to maintain that patent because of the cost of the yearly maintenance fee. This last year, the BMP2 with collagen sponge, which was my idea, collagen sponge, sold over $1 billion last year, $1 billion. So if you do the calculation, Case blew a huge amount, spent all the money to write the patent, spent all the money to file the patent, didn't maintain the patent because of the annual fees, which is nothing. Okay, and so therefore, you can imagine that I mentioned this to Joe Jankowski on several occasions. So there's 2 take-home lessons. One is this patent, although somebody else beat us, as I'll tell you in a second, to the BMP cloning that gene, we had a blocking patent for the delivery vehicle. So again, the point that was made this morning, sometimes you patent something for one reason, and by 10 years later, when that comes to the market, one of the claims becomes hugely important for the use of that material. So you don't know, no crystal balls, what your claims are going to be 10 years down the line. So there was a company called Genetics Institute in Cambridge, cloned BMP2. So I knew we lost the race because we didn't even have the amino acid sequence for CSA. So what happened at that point was that this light bulb went off in my head, which is that I've been taking these kinds of molecules and putting it on embryonic cells and getting them to become cartilage and bone, okay? And I remember Marshal Yours' first experiment. He jams this demineralized bone into an adult mouse muscle and he gets bone from that. So I said there must be in the mouse, adult mouse, other kinds of stem cells comparable to the mesenchymal stem cells that I got from the chick embryo. And that was one of those magic moments, and we went after this cell, which I called a mesenchymal stem cell. And we generated this picture, and I'll show you the origin of this picture, which is there's a paper by Maureen Owen in a very obscure review article where she puts the hematopoietic system, and we know a lot about these details, with this mesenchymal system. And from the stimulus of that paper, and this is before computers, you have to understand. So this is all done on a manual typewriter by me, hunting and pecking. To draw this fabulous picture on a typewriter is ain't easy, I have to say. And I sent it to 20 of my esteemed colleagues in the field and said, what do you think about this idea? So I won't share with you their scholarly responses because they weren't so scholarly. So this is the original diagram, the actual picture of the original diagram. I still have the piece of paper in my office. And this is from the offering document when Osiris tried to go public. So you can see when you're doing Wall Street, you know, it really bumps up but the basic information's identical. And actually this is the drawing that the Case Western Reserve art students don't like my blue slide, which I of course think is fabulous. This is their rendition of the same figure done to please other people, not to please me, clearly. But it's the same basic information about the multipotency of mesenchymal stem cells. And so I want to just put this in context for all of you, is that there is a National Center of Regenerative Medicine. It's run by Stan Gerson, and you'll see why that's important. But the Skeletal Research Center, there's an Armed Forces Institute of Regenerative Medicine branch run by George Mushler. Mary Laughlin started a cord blood center, and George Mushler and I started the Clinical Tissue Engineering Center with state money. So mostly of this was started with state money, but now there's this national center, so there's a huge number of people in Cleveland working on various stem cells, not only mesenchymal stem cells. So this is the hypothesis slide. This is Steve Hainsworth, who was a postdoc in my lab. Hainsworth's now associate professor and associate dean of the College of Arts and Sciences, so I now report to Dr. Hainsworth. It's the ultimate irony. We're wonderful close friends. And he and I developed a way of isolating from bone marrow these mesenchymal stem cells. And so you put them on a petri dish, and individual cells divide like crazy under the conditions we've set up, and you get a colony. And you can count these colonies. So you could count the number of stem cells in an individual, and I'm going to ask you to memorize this slide because it's hugely important. 1 in 10,000 of these marrow cells in a baby is a mesenchymal stem cell by this colony forming assay. By the time you're a teenager, it's down to 1 in 100,000. By the time you're as old as Gabrielle, you're in bad shape. That's why she befriended me. Needless to say, I'm so old that I'm way down here on the bottom side of this chart. And that has to do with your regeneration potential. And this business of cutting off your finger and not being able to regenerate is a stem cell availability issue as part of the problem. So so this is from bone marrow. I want you to memorize that. If there's a few minutes at the end, I'll tell you the data is correct. there's a completely different explanation for this particular data set. So the multipotency in tissue culture, we can get these cells to grow. We put them in these different medium. We can get bone, cartilage, or fat in culture, and that's called an assay for multipotency. So here are the 4 basic patents that describe the isolation process, the use of this for treating, quote, connective tissue disorders, the way we uniquely identified mesenchymal stem cells by cell surface markers that we develop monoclonal antibodies for. So this is the core set of patents that Case Western Reserve generously, with no royalties, transmitted to Osiris in a technology transfer arrangement. This is Stan Gerson. In his youth, he's lost most of that hair. He's now in charge of the Cancer Center. Gerson is almost single-handedly responsible for this technology translating to humans, because I took this technology to Gerson and said, for bone marrow transplantation, MSCs can make bone marrow stroma. When you do a bone marrow transplant for a cancer patient, this could add value to what you were doing. He bought that idea. And then, so if you buy that idea, you have to take this technology and do a clinical trial. So we did the first investigator-initiated clinical trial, and it took us 1 year to get this through our institutional review board, and if it wasn't for the patience of Gerson, I would have quit the process. They asked the most obnoxious, detailed questions that were, for scientists, were trivial and worthless. Gerson been through the process, held my hand, said, Arnie, take it easy, we'll get it through the process. And again, if it wasn't for Gerson, would never have done this. I would have blown my cork several times. So he got the help of Hillard Lazarus, who ironically now is the institutional head of the IRB. So Hillard's been involved in this, and actually the first publication of this first infusion of mesenchymal stem cells for safety studies, and Hainsworth and I did the cultures of these cells in my research lab, which of course would never work now for various reasons. But let me assure you that my armpits were like faucets when I was doing this, because this was not science. This was going into patients, and the stress on us to not contaminate the cultures to get good product, gigantic. And so that was very illuminating for a theoretical scientist to have to actually do something in a practical sense, and at the scale where there's about 50 million cells for us in those days was gigantic. And so that was a gigantic amount of work. And what we showed was you could infuse this into patients with no harm. And actually there are now clinical trials, I won't go through them, that show actually these cells add value to the bone marrow transplantation. For breast cancer patients, there's a completed clinical trial that says that these cells inhibit the immunocrashes that the patients have, and some of the downsides of bone marrow transplantation. Bone marrow transplantation is, with chemotherapy and radiation, they kill all your hematopoietic stem cells. So those 15 million cells that are dying every second don't get replaced unless you put back a hematopoietic stem cell. The mesenchymal stem cell helps that cell engraft and do its thing. It doesn't do it the way we thought. It doesn't build the scaffold. It does it by drugging the site. There's no data for that. So here's the Gerson Hainsworth patents for enhancing bone marrow transplantation. So these are actually revolutionary patents in adding another cell to a bone marrow transplantation, a purified culture expanded cell. So tissue engineering, you take a bone marrow aspirate, you get the cells in culture, we can freeze them down, thaw them out, and we published data on all of these tissues. So in 1990, again, the patents were all written. This is called the classic academic disconnect, okay? So I wrote my first grant on mesenchymal stem cells, right? And what did NIH say? I have this in writing. There's no such thing as a mesenchymal stem cell. And of course, what did I say? Is that I wrote 4 patents, there better be something called a mesenchymal stem cell. The grant never got funded, I have to say. And again, it's when you do something revolutionary, not only does it take time to get to market, but it takes time to educate your esteemed colleagues, which is a problem for the Genome Project, a problem for a variety of other technologies that you'll talk about. So I started a company called Skeletech, which embodied the mesenchymal stem cell technology. And here's our mission statement. You can see this is a typewriter. This is a photograph of the typewritten script. And again, this is against all dogma, regenerating tissue. And here's the picture that wasn't the pretty blue picture that eventually came, but it was this picture, and that's the basis of starting Osiris Therapeutics. So I used to talk about all of this tissue engineering stuff that mesenchymal stem cells do, but what mesenchymal stem cells really do is that they pour out gigantic quantities of molecules. And so these are called bioactive factors. One group of them are immunosuppressive. This is the mechanism for my more academic audience of how actually the mesenchymal stem cells stop your immune system from surveying the damaged tissue. And so there is an absolute molecular mechanism behind this activity. And this is what allows Osiris Therapeutics to sell allogeneic, somebody else's stem cells frozen in a bag for use in infusion technologies into people with various maladies. This is the young and very clever new CEO of Osiris Therapeutics, Randy Mills, who actually stopped the academic work at Osiris and started doing clinical trials, which changed the face of this technology. And his business plan is very easy. You get an aspirate from a young donor, you expand the cells by the technology so cleverly transferred by Case Western Reserve University, and you get 10,000 bags of cells that you're going to infuse into people. And this is the pipeline, the clinical trials that are now going on, and I'll go through these rather quickly. And their worldwide partner is now Genzyme Corporation, a pharma company. And this is an old slide. Over 120 in 2010, over 120 clinical trials, registered clinical trials, going on with MSCs. That's not to say the couple of hundred other clinics that are doing medical tourism that are using the same technology, identical technology. And so Osiris has clinical trials in over 200 centers worldwide. And actually the next slide is not a Photoshop slide. This is a picture I took personally with my camera in Cape Town, and it's clear these guys are shipping these things all over the world, and that's how they get there. And if you want to talk about and these are real pictures, so I mean they're not Photoshop pictures, honestly. So talk about safety and regulatory. That ship, this is from the Wall Street Journal in August 10. I mean, I took this picture off the Wall Street Journal website. This ship ran aground in the Indian Ocean and was clearly delivering its material locally. And the last slide is that you can now go on a cruise ship. This is in New York Harbor, a cruise ship actually, and get your infusions right while you're overeating in New York Harbor. So MSCs, you now know that MSCs is just not a Kaplan invention. So because of the time, I'm only going to talk about a couple of these clinical trials. One is steroid refractory graft versus host disease. Cancer patient gets nuked or chemotherapy, and they get somebody else's bone marrow. And that somebody else's, the graft, somebody else's bone marrow starts rejecting their body. So their guts fall out. Your GI tract is completely disabled. You have huge skin lesions. You're bleeding. Your liver is shut down, and it's called graft versus host disease. Here's kids. This data just knocks my socks off. Kids who were certified 3 to 5 days from dying of graft versus host disease. And on a compassionate use basis, Osiris gave these kids multiple doses of mesenchymal stem cells. So if I had Gabrielle's mesenchymal stem cells infused in me, I would assume the first one, my arm would clearly tingle. Okay, the second infusion, I would figure that my arm would blow up from the rejection. The third infusion, I would expect my arm to fall off my body from the rejection. Up to 20 separate infusions into these kids, no adverse events, 0 adverse events. The immunoregulatory capacity doesn't allow the immune system to see them, and they do their business. And the first thing they do is they get the gut, the GI part of the graft versus host disease, into shape in a matter of days. So it shuts down this inflammatory aspect, and that's why eventually Osiris did Crohn's disease. Just to show you this is not a hype for Osiris, Katarina LeBlanc in Sweden published her own data on this, and it's the same story. It's different doses, multiple doses, either on identical SIBs or completely mismatched recipients, and the same very positive results for graft versus host disease. Crohn's disease, this inflammatory bowel disease, this data from their phase 2 clinical trial is spectacular. There's a dose-dependent improvement in the clinical score of these patients. It keeps going up 150 points, 120 points. On that scale, there's nothing that's being sold, no drug available that could do that in this kind of time period. And again, it's multiple infusions, at least 3 infusions. I'm not going to go through, this is our data on asthma, clearing up the lung, and what I want to do is I want to talk about, I'll talk about heart attacks. You get a heart attack. This is a model for heart attack. You basically occlude one of these major arteries, and that part of your heart is not going to get nutrition or oxygen, and you're going to get a huge inflammatory response, scarring, damage, and actually what happens is because this part scars, this part blows up, becomes hypertrophic, and you get electrical discontinuities, and that's what you die from. So there's a mechanism that MSCs work, but here's the phase 1 clinical data from Osiris's trial within 48 hours after getting a primary infarct. These patients were infused with allo, somebody else's mesenchymal stem cells, fewer arrhythmias, fewer premature ventricular contractions, prompt return to heart rate after exercise, and the unexpected finding was an improvement, 3 to 4-fold improvement in lung function. So people get heart attacks, are smokers, they have other problems. The MSCs are stupid. They go to sites of damage and injury and do their thing. So, I mean, this is their data. So this is what I call trophic, and that's a real regeneration. And so I'm just going to show you one last experiment. This is the dapper Dr. Bob Miller, who's in the neurosciences department. He works on multiple sclerosis. He works on neural stem cells. And so a neural stem cell can differentiate into neurons, astrocytes, which are sort of the fibroblast of the nervous system, and oligodendrocytes. Oligodendrocytes are the cells that wrap your axons with myelin. Okay? So being an enormously generous soul that I am, I took the medium that I was going to throw away from the mesenchymal stem cells that I'm expanding in culture, and I shipped it off to Miller's lab, and he put it onto a neural stem cell culture. You put these neural stem cells in culture, and they go to these 3 different cell types, and you can quantitate them. And what Miller and his colleagues found was that there was a 10-fold decrease in the astrocyte differentiation and a 4-fold increase in the oligodendrocyte differentiation. So that was spectacular. So that made us think, let's do this animal model. It's called the Eae Model. You put a peptide that is from myelin in the animal, and the animal develops MS. Its immune system attacks its myelin, and this is a clinical score, which is by the time the animal, and these are groups of 10 animals each, so each of these points is 10 animals. By the time the clinical score, it can't walk or feed itself. So all these animals are kept alive by technicians doing this by hand. Right at that point, if you give these animals, these are mice. If you give them 1 million human into mouse, human mesenchymal stem cells, what you see is within 2 weeks, 3 weeks, these mouse walk away. They're totally normal. They're cured. And what happens is, I don't have the data here, but what happens is that the MSCs go to the sites of injury. They stop this inflammatory attack on the central nervous system, and those axons which have been unmyelinated are remyelinated because the MSCs take the neurostem cells that are intrinsic to the animal. They differentiate into oligodendrocytes and rewrap those nerves. So it's the host curing itself with a little help from MSCs. So I call MSCs a site-regulated multi-drug delivery vehicle. It's a drugstore for sites of injury, and it's the mechanism that we can now understand, because we understand that the MSCs are sitting there normally as pericytes, that they come off the blood vessels at sites of injury and produce these molecules. These molecules stop the inflammatory response, the host inflammatory response, and set up this regenerative microenvironment. And so all of this has to do with all of these things which I've listed previously. This is the mechanism. And for all of these, and again, all of these molecules, it's not one molecule, it's many molecules. And so this mechanism is what you're augmenting by sending mesenchymal stem cells through the bloodstream by infusion to these different sites of injury. And this is going to require a completely new set of patents. As a matter of fact, what kills me, of course, is I've had to change my brilliant hypothesis slide so that now I have to put another cell up there, because that's the business end of the normal process that we're trying to organize in patients. And this slide is a slide of your blood vessel density in your body. When I cut myself, shaving takes 2 or 3 days. Young guys cut themselves, next day it's fine. It's blood vessel density. I can take a skin biopsy of every person in this room, and I can tell you how old you are or whether you have diabetes by the blood vessel density. And that's what this curve is in bone marrow. What controls blood vessel density? I don't know, but I'm working on it. Clearly I have a personal interest, but it changes the focus of research, and it's also now changes the focus of the way entrepreneurial thinking structures patent portfolios, because it's the vasculature which is the driver, not the MSCs for healthcare. And these are all of the clinical opportunities that are now being explored using this technology. And what's very clear is that 20, 30 years ago, when I started calling these cells mesenchymal stem cells, I got into huge trouble because the dogma of the day said there's no such thing. So what I say at every lecture is that you should call these cells Arnold Kaplan cells, and you won't get into any trouble. And of course, none of my papers have been published that way because of the snooty journals that I have to publish in. And like all hypotheses slides, this is hugely naive. And what we now know is that we can take fat cells And in culture, we can get them to be quantitatively bone-forming cells. This is called plasticity or transdifferentiation. There are lots of names for these. And so we're trying now to really figure out the complexity of that diagram, which is enormous. And the control elements are likewise enormous. And again, there will be patents that derive from this. And it means also that you can take fat cells and make bone from them, or cartilage, etc. So it opens up new avenues. And this is one of the last slides. This is a Photoshop slide by one of the wise guys in my laboratory that appeared on my door one day, and it's to remind me to tell you that you've clearly had a highly prejudiced, focused MSc lecture. Clearly, there's lots of other technologies and ways of looking at this. What really gets me PO'd about this slide is that it was a picture, a real picture and this kid was selling newspapers, and what was on the banner was that the Titanic sank. So I hope it's not prophetic. We teach a course in May, which is called Cell-Based Therapy and Tissue Engineering, where I bring in a lot of people who use these tissue engineering technologies from both a scaffold side and a cell side, and many stem cell experts to talk about the science of this. And actually, there's a business course that we teach in July where I bring in the CEOs, the chief scientific officers, to talk about the business aspects of this. And I bring in a lawyer to talk about patenting strategy, because clearly it's seminal to everything we do in the business sector. So it's clear that all my research has been generously supported by the National Institutes of Health, and there's a cadre of people in the skeletal research center that really provide the data, for whom I owe a debt of gratitude. Thanks. Questions, if you dare. Yes, ma'am.
This is a layperson's question. So, thank you. Given the vast number of people on Medicare and elsewhere who are getting hip replacements, which I understand is not basically because there's something wrong with their bone, but because there's something wrong with their cartilage. So what point can we just inject cartilage the way you inject jelly in an Amy Jo donut and be done with it?
You know, within the next 5 or 10 years, there will be a variety of solutions to cartilage damage issues. The problem is the following, which is that if we make a piece of cartilage in tissue culture and we put it in your joint, the real medical problem, molecular problem, is that new cartilage has to integrate with your old cartilage. And that's the tricky part, because we don't have effective ways of doing that. And then even if it integrates at the edges, the new cartilage is going to have a different mechanical property than your old cartilage. So there is a point where if you're a 20-year-old or a 30-year-old and you damage your cartilage, there are no mechanical fixes for that. You need to get a plug put in there. But if you're over the age of 50, your cartilage has so degenerated because of age and your genetics that you're better off doing a prosthetic device. There's a secondary problem for which MSCs have a clear advantage, which is there's a period of time when you have knee pain and you have osteoarthritis, single joint problem, not rheumatoid arthritis, osteoarthritis. And there are some experiments by Osiris and by others that show that if you just squirt some MSCs in your joint, actually you stop this inflammatory activity, and actually the outcome scores are pain scores. So there are going to be some people who have damage to their joint, which is different than a hunk of cartilage coming out, that have severe pain, that pain can be managed by an MSC injection or multiple injections. So there are going to, so again, it's a multi-drug approach, very different than current therapies. What doesn't happen is the cartilage that's damaged doesn't regenerate, because you don't have that potential at ages above the age of 20, 25. It used to be in my generation, you peaked out, your skeletal maturity peaked out in our early 30s. The new generation, the current generation, is in their mid-20s because of nutritional aspects. You see the kids are bigger, they're taller, they're healthier, they come to sexual maturity earlier, and their skeletal maturity is earlier. So this is a nutritional issue. So what I tell people is my father lived to be 97, my mother lived to be 93. None of them had joint replacement surgeries, so the chances are I'm not going to do that. I'm going to die from Alzheimer's or other diseases. I know what my problem is, and you may be experiencing some of the side effects by listening to me talk so long. So, but there are no quick fixes. It's one of the real problems in healthcare delivery in the United States is that the average individual on Medicare or Medicaid thinks that they can abuse their body, go to the doctor will give you a quick fix, and you go home, you'll be okay. Doesn't work that way. So these are complicated. Cell-based therapies are complicated, costly therapies, and they're going to work for some people, and they're not going to work for others.
I see Alzheimer's as the next epidemic that we'll be facing.
Actually, I can't hear you. Is that on?
Can MSC help with Alzheimer's?
Probably not. But Alzheimer's, it's a real problem, a verbiage problem, which is that for insurance purposes, again, for economic purposes, a large number of maladies are now clustered under the Alzheimer's logo. So my father, quote, had Alzheimer's. He had senile dementia. Senile dementia has a different sequelae of complications that build up to cause that, very different than Alzheimer's. So if somebody has Alzheimer's at the age of 40 or 50, that's Alzheimer's. If somebody is characterized who has senile dementia, that's a vascular and has other complications. The treatment plan for those people in the terminal phases is identical. So you have to be careful about, and it has to do with rheumatoid arthritis and other maladies. What we're trying to do in the medical community is design diagnostics to help us characterize patients so that we can prescribe proper treatment plans for them, including proper drugs. And that's part of what the genomics activity is centered on. If you know the gene composition of an individual, can you predict whether a drug will be effective or not effective for that individual based on that kind of knowledge base? Eventually, our hope is that we'll be able to be smarter about prescribing drugs and dosing based on the genetics of the individual, as well as a better understanding of what their malady is. So again, the rate limiter here is diagnostics. In the healthcare field, it's the hottest, newest area that's being explored, and again, it's trying to correlate that kind of measurements with what your genome is all about. So it's a very complicated time period. The next 10 to 20 years will show a very different relationship between information obtained from patients and healthcare protocols. Yes, ma'am.
question about your preclinical research on the introducing human MSC cells into mice.
Yeah.
I was wondering, what were the cells that were regenerated and resulted from the human cells? Were those human cells or were they mouse cells? And if so, what if they were mouse cells? What happened to the human tissue that was inserted? And would you do this on animals? Would you put human stem cells in for brain regeneration in animal models?
So again, you're mixing apples and oranges. So the MSCs that we put in are pouring out huge quantities of molecules. So, and those molecules are having therapeutic effects on the host. So the host tissues are the ones that regenerate. So there are no human tissues in those mice, okay? The mesenchymal stem cells don't last forever in those animals, and eventually they go away. How they go away, why they go away, we don't know. We're certainly working on that. But the startling fact is that your cells, your MSCs, in my body would have a therapeutic effect. So that's 10 years ago, I would have told you, don't do that for ethical and for scientific reasons. We now have enormous data on this that says it's safe. So it does the first rule of medicine is do no harm. So whatever procedure you do, you can't make the patient worse. So in this case, when we put human cells into a mouse, it's actually a better preclinical model than putting mouse cells back into the mouse, which is, from a scientific sense, the proper experiment. But by putting human cells in, we're actually testing their efficacy against a particular set of circumstances in the animal. And we project that that's the way it's going to work in human. The only way to prove that is to do human clinical trials. And human clinical trials are set up in phase 1, phase 2, phase 3, safety first, then safety and efficacy, and then only efficacy, real efficacy in large numbers of patients. So the important aspect is that preclinical models are a way to argue to the FDA that you can do a safety study. After you get the results from a safety study, you can then do a safety efficacy studies, phase 2. And then eventually you do a clinical trial that would allow you to label a certain procedure or drug that it's efficacious for X. And so, again, the step, what we do with animals is a scientific exploration.
I'm just wondering if you have any ethical concerns about chimeric.
Not if it does no harm. I mean, and as a matter of fact, what I'm trying to do is, again, there's some considerable resistance, is if you can use human cells in an animal model, I want to use human cells because they're what's going into patients. And I want to test that. If it does something really bad in an animal, I want it done bad in that experimental animal. I know it's not a nice thing relative to the animal, but I'd rather have an experimental animal suffer the consequences than a human. And so the rule in the FDA, the rule in all drug and device and other appliances for medical care is you have to have animal data. And it's the first level of testing whether something has a chance of being efficacious. I'm supposed to cut and say we're going to take a short break and then we'll have the next speaker.